All right, nutrient digestion. In order for us to absorb our foods into our bodies, they have to be broken down. And they are broken down into smaller parts, smaller pieces through digestion. So we have mechanical digestion and chemical digestion. So in mechanical digestion, our teeth through chewing, the muscles through moving it around, breaks that food down physically and increases the surface area of it so that chemicals can contact it. So that would be chemical digestion, where we use chemicals to break down the rest of that food into even smaller components. So we have carbohydrates, right? We have proteins, and we have lipids. And those are the things that we consume that have to be broken down and absorbed. Those are the nutrients. So carbohydrate digestion, and I'm talking specifically about chemical digestion now. So the chemical digestion of carbohydrates, of course, starts in the mouth. So in the mouth, when you are chewing up your breads and sugars and things like that, your mouth contains an enzyme within saliva called salivary amylase. And that salivary amylase, starts the breakdown of the big carbohydrate molecules into smaller ones, so smaller polysaccharides. Okay. Once those polysaccharides end up in the stomach, carbohydrate digestion stops there. Once it gets into the small intestine, that digestion of carbohydrates resumes. So in the small intestine, where we get these polysaccharides, we get pancreatic enzymes. So pancreatic amylases, amylase, I'm a terrible speller. So pancreatic amylase, which breaks those polysaccharides down into their disaccharides. And we went through this in AP1 when we talked about in chemistry these different nutrients. And a disaccharide is two sugars linked together, and there are 3 of them. So the body is going to break the polysaccharides down into these 3 things. So maltose, lactose, and sucrose. So maltose, when it is broken down, because what will happen is this maltose, lactose, and sucrose then will get right next to the wall of the small intestine, and there'll be special enzymes for it that will break them down into their monosaccharides. So there will be maltase, lactase, and sucrase. And those will break the maltose, lactose, and sucrose down into their monosaccharides. So glucose, galactose, and fructose. So once those are broken down into their monosaccharides, then they will enter the small intestine cells and then be processed into the blood. So glucose and galactose enter the cells through secondary active transport, and fructose enters those cells through passive transport. So basically diffusion. Proteins, their digestion, their chemical digestion starts in the stomach. And in the stomach, we have pepsinogen that contacts the hydrochloric acid of the stomach, and that gets activated into pepsin. And pepsin starts the chemical digestion of proteins by breaking those large molecules, large proteins, down into smaller peptides, polypeptides. So once that occurs, then they enter the small intestine, where they are worked on by proteases secreted by the pancreas. So pancreatic juice has all of these enzymes in them. So these proteases, the amylases, all of that to break down these components. So once these peptides are broken down into smaller peptides, then they will make it over to the wall of the cell at the lumen edge, and these peptides will be broken down into tripeptides, dipeptides, and amino acids. So a tripeptide has 3 amino acids linked together by peptide bonds. A dipeptide has 2 amino acids linked together by peptide bonds, and then an amino acid is just itself. Okay, so those 3 forms of proteins can enter the blood, enter the cell, the small intestine cell, and then enter the blood. Lipids are a little different because they have to be processed first. So lipids have to be emulsified. So they go through this process of emulsification. And the chemical needed to do that with lipids is called bile. So basically what happens is you've got this lipid and you've taken in this fat, and it's in our watery environment of our digestive tract, and it's just going to form this big old fat blob. And a big old fat blob can't be chemically digested well by lipases. Those are the enzymes that digest fats. But the lipase can't get to it because it's all encompassed in this big fat blob. So the bile, what emulsification does is the bile then breaks that blob down into many little tiny blobs. And that makes the surface area much larger so those lipases can contact it. So we're going to go through each one of these on a separate video: carbohydrate absorption, protein absorption, and lipid absorption.